This passage is adapted from Maddie Stone. We have been wrong about lichen for 150 years. Lichen are complex organisms composed of algae and fungi. Hundreds of millions of years ago, a tiny green microbe joined forces with a fungus and together they conquered the world. It's a tale of two cross-kingdom organisms, one providing food and the other one shelter. And it's been a touchstone example of symbiosis for 150 years. Trouble is that story is nowhere near complete. A sweeping genetic analysis of lichen has revealed a third symbiotic organism hiding in plain sight, alongside the familiar two that has eluded scientists for decades. The stowaway is another fungus, a Basidiomycete yeast. It's been found in 52 genera of lichen across 6 continents, indicating that it is an extremely widespread, if not ubiquitous, part of the symbiosis. And according to molecular dating, it's probably been along for the ride since the beginning. I think this will require some rewriting of the textbook, said Catherine Aim, a mycologist at Purdue and co-author on the study published in Science. Toby Scribble, who led the new analysis, has been studying lichen in one way or another for most of his life. He grew up in northwest Montana, where the shrubby, rubbery organisms are a ubiquitous part of the natural landscape. But when he started to get serious about lichen research in grad school, he hit a roadblock. Lichens are nearly impossible to resynthesize in the lab, he said, explaining how the colonies take a long time to grow and the conditions needed to induce symbiosis are not well known. Unable to rear their test subjects in controlled environments, lichen researchers have struggled to perform basic experiments that could shed light on the roles of the different symbionts. But recent advances in metagenomics, tools for extracting and sequencing DNA from environmental samples, no culturing required, offer a new way in. This approach caught Scribble's attention when he learned something very strange about Bryoria, a lichen found throughout conifer forests of the western US and Canada. Bryoria have a long and storied cultural significance, he said, explaining how certain Native American tribes relied on the lichen as winter survival food. There's also evidence that first peoples would remove the more light-colored ones and wash them so that certain substances wouldn't make them sick. Those substances include a toxin called vulpinic acid. The lichen that produces it, Bryoria tortuosa, can be distinguished from a non-toxic cousin, Bryoria fremonti, on the basis of its yellowish hue. But a few years back, when a group of biologists at the University of Helsinki tried to uncover the genetic basis for this difference using a targeted approach called barcoding, they were stumped. They found that the toxic and non-toxic forms were identical, at least in the known parts of the lichen, and they didn't really study it further. We looked at that and said that this is a classic question you could go at with genomics. When Scribble and his colleagues analyzed Brioria's RNA, the messenger form of DNA, they discovered something amazing. We found that there was this third thing riding along in every single sample. He said, referring to the previously unknown Basidiomycete. At first, the researchers worried that the extra RNA sequences could be contamination, a common pitfall of genomic research. And so they decided to see if they could find the Basidiomycete in other lichens too. We found it in everything, Scribble said, from Alaska to Ethiopia to Antarctica. It was always there. The final proof that this was not an elaborate hoax came when the researchers developed green fluorescent markers that attach to specific RNA sequences in the Basidiomycete and blue markers that attach to complementary RNA sequences in the other fungus and Asomycete. Sure enough, when they added these markers to samples of lichen tissue, the cells of a hidden fungal partner glowed under the microscope. We can't be certain that the second fungus is present in all lichens. Scribble's study only looked at the lichens in the Palmiliaceae family, the most widespread and successful group on Earth today. But the entire lineage is vast and ancient, and it's possible that some groups split off on the evolutionary tree before the Basidiomycete arrived on the scene. In context, the main purpose of the sentence in line 6 to 7 is to So what is that sentence? Trouble is that story is nowhere near complete. Is to advance the argument that scientists still have much to learn about the interactions of organisms across kingdoms, challenge the assertion that algae and fungi are especially dominant types of organisms, suggest that research into lichens has generally been proceeding at too slow a pace, introduce the idea that the most widely accepted understanding of lichens is flawed. So it would be D, right? Because we are not talking about interaction of organisms across kingdoms. This is to do with lichens in particular. We are not challenging the idea that algae and fungi are dominant in general. This is, again, we have to look at an option that talks about lichens. And the idea that this understanding is incomplete is not because research is proceeding at too slow a pace. As used in line 8, sweeping most nearly means a sweeping genetic analysis of lichen has revealed a third symbiotic organism. So a detailed, right? Extensive, something that is done on a large scale, right? So that would be the meaning of sweeping. Indiscriminate doesn't fit the context here. Indiscriminate would mean without thought. Spacious has a physical context, and unconditional doesn't fit. In lines 9 to 11, the phrase hiding in plain sight and the words eluded and stowaway mainly serve to. So, hiding in plain sight, allowing the familiar to, that is eluded scientists. The stowaway is another fungus, right? So, hiding in plain sight, eluded scientists, right? So, these words, what is their purpose? Identify the challenge inherent in using genetic analysis to isolate this yeast, characterize this yeast as a component that was undetected even during studies of lichens, highlight traits of this yeast that led scientists to misclassify it as a different fungus, hint at the likelihood that the presence of this yeast will be visible in additional genera of lichen. So this is B, right? Because we are saying that this was present all along. And we also know that molecular analysis has revealed that molecular dating has revealed that it has been present in lichens all along, but it has eluded scientists, right? So it was there, it was a component that was undetected even during studies, right? This is not a challenge inherent in genetic analysis. In fact, genetic analysis finally told us about this. It was not, there's no misclassification here. It's just that it wasn't detected. And additional genera of lichen is something that is not even relevant at this place, right? The passage indicates that molecular dating allowed Pribble's team to determine that this yeast has likely always been present in lichens in the Palmylaceae family, that the algae found in most locations around the world today have an ancient lineage. The organism with which this yeast first developed a symbiotic relationship, the point in time when other common lineages of lichens branched off from the Palmylaceae family. So this is A, right? Because we know that Scribble's study only looked at the lichens in the Palmylaceae family, the most widespread and successful group. And molecular dating said that this fungus was present right from the beginning, right? The fact that most locations, most algae have an ancient lineage, or that there was branching off, this is not the purpose of the molecular dating that was done by Scribble's team. And the organism with which it developed a symbiotic relationship, that's already known. So that was not the purpose. A main purpose of the fifth paragraph, lines 28 to 35. So this one, right? They are nearly impossible to resynthesize in the lab, explaining how the colonies take a long time to grow and the conditions needed to induce symbiosis are not well known. Unable to test subjects in a controlled environment, these researchers have struggled to perform basic experiments. So this tells you the trouble or the problems with testing. Question the methodology scientists have used in most studies. Highlight the amount of time required to cultivate lichen colonies. imply that scientists must find a new way to synthesize lichens, explain some of the difficulties of conducting research into lichens, right? So these are the traditional difficulties which were solved with genetic analysis, right? It's not about questioning the methodology. That's not the problem. The methodology is fine. It's just that it takes a lot of time. It takes a lot of time, but that's not the only point. It's the point of the paragraph is to highlight why you needed a new method to study them. And again, that implication is the fact that they need to find a new way. They already have, right? So it's not that it's something that needs to be done. It's implied, right? But the main purpose is to explain the difficulties. Based on the passage, the author would most likely characterize Pribble's team's conclusion about the RNA found in Bryoria tortuosa and Bryoria fremontal lichens as somewhat. The author would characterize their conclusion. Okay, so let's first mark the line numbers. Let me just open this again. Okay, so we are looking at words like ironic, puzzling, satisfying, and frustrating. 54 to 57. 54 to 57. Then we have 58, 61, 64, 69, 58, 61, 64, 69, and 70 to 72. Okay. Okay, and we are looking at evidence for how would the author characterize the team's conclusion about the RNA found in these two lichen. Okay. But a few years back, when a group of biologists at the University of Helsinki tried to uncover the genetic basis for this difference, they were stumped. Okay. They found that the toxic and the non-toxic forms were identical, at least in the known parts of the lichen, and they didn't really study it further. When Pribble and his colleagues analyzed Bryoria's RNA, they discovered something amazing. We found that there was this third thing riding along in every single sample. At first, the researchers worried that the extra RNA sequence, okay, so it has to be this option, right? Because this gives you direct evidence for Spribble's excitement. So Spribble's team's ironic, puzzling, satisfying, and frustrating. Okay, so I like this option. Let's just read it. Because the team's previous attempts to provide genetic proof in lichen samples had been inconclusive. The team's attempts to isolate and identify a genetic difference between the two lichen species revealed a commonality between them instead. Okay. So let's just go back and read that. They found that the toxic of the were identical. Yeah, and they didn't really study it further. Okay, no. We are talking about Spribble, not the people before Spribble, right? So it is, I don't think it's A then, because the team had assumed that the presence of acid would prohibit the, no, it's not about, seems unlikely that the team will be able to find the same, no, okay. Okay, it's interesting. I'm just trying to figure out, this was also Spribble's team, right? Those substances, University of Helsinki, where was Spribble working? Okay, no. So this was another group. They found that the toxic and non-toxic form, and they didn't really study it further. This third thing. Okay, so this was, yeah, okay. So they were looking for something else. Right. Yeah, so it is not A because this part was the researchers before Spribble, right? They discovered a commonality between them, but they didn't study it further. But this team was able to provide a genetic proof of find that evidence, right? So yeah. Which potential finding would provide the best support for the passage's main conclusion about lichens? Okay, and this is not evidence. Researchers determined that the genetic markers particular to the algae in lichens are not susceptible to contamination. They collect more evidence to prove that the acomycete in lichens has a unique RNA sequence. Okay, I'll come back to this question. Let me just look at 30 and 31 first. Which conclusion about this yeast is best supported by the passage? Okay, let me mark the line numbers first, and then we can look at these options. So the first one is 28, 31, 39, 42, 28 to 31, 39 to 42, 77, 82, 89, 92. 77, 82, 89 to 92. Okay. And what are the options that we are working with? Which conclusion? It may have been absent in distant ancestors of the lichens. It may have initially developed as a result of lichen contamination. It may be impossible to reproduce from cultures. It may be a necessary component of symbiotic. So I think this would be A, right? Because this evidence, the entire lineage is vast and ancient, and it's possible some groups split off on the evolutionary tree before the basodiamycete arrived on the scene. So the idea that this yeast is, the idea that this yeast was always present is not supported. So yeah, and this is what option D is saying. Okay. Now, if researchers determine, okay, I just wanted to read that part of the passage again because this part is where the answer will come from. At first, the researchers worried that the extra RNA sequence could be contamination. The final proof that this was not an elaborate hoax was when they developed green fluorescent markers. Other fungus was acomycete. Okay. When they added these markers, the cells of a hidden fungal partner glowed. Okay. Researchers determined that the genetic markers are not susceptible to contamination. They collect more evidence to prove that this has a unique RNA sequence. They learned that this matched those of lichens from other families. Researchers discovered are present in lichens outside. Okay. So see, the fact that the genetic markers are not susceptible to contamination, that's not relevant because we already showed that they are different, right? The two fungi are different. This has a unique RNA sequence. Okay. So I'm going with D but I don't know if this is the conclusion that they wanted to prove, right? Because the conclusion is not that they are present everywhere. They're at least present in the Palmoliaceae family. So yeah, I'm not really happy with D either. Okay. Let me just see what it was saying here. They found that the toxic and the non-toxic forms, they didn't really, okay, that was when this team analyzed, they discovered, we found that this was this third thing. And so they decided we found it in everything. The final proof, sure enough, yeah. So this is, it's not A. RNA sequence. That has already been proved, right? It does have the markers were different. Bryoria's DNA sequence. I'm really not sure about this. So yeah, this question, I think it should be between C and D, but not sure. Okay, let's grade this 22. 22 DCBAD. DCBAD, 27 ACDAD, 27 27 ACDAD. Okay, so this is A, C. Oh, this was D, right? So this is the conclusion that we are looking at, AD.